I'd like to introduce our speaker for this evening. Catarina Miranda is the kind of young person who gives hope to old native plant lovers like me. She's studying biology. She's an ecologist and an avid naturalist. She's currently working toward a master's degree in biology at UTRGV, where she is a graduate research assistant in Dr. Christofferson's ecophysiology lab. Her research focuses on Tamaulipan thornscrub, and that is the major biome of South Texas. And she's researching how to restore it successfully. Catarina was a recipient of the 2024 Native Plant Project's Native Plant Propagation Grants. She runs a small backyard native plant nursery, and she has brought some of her plants to sell here tonight, over here on this table. So now let's welcome Catarina Miranda.
Hi. I guess I was already introduced, but I'm going to introduce myself again. My name is Catarina. I also go by Cat. And I actually have a slide here where I'm kind of talking about myself a little bit, but first I'm just going to tell you what this presentation is. It's called How Does a Forest Sustain Itself? The Role of Microhabitat and Succession on Recruitment in Tamaulipan Thornscrub. The titles of scientific papers tend to be mouthfuls, which is why it's not like a super intuitive title, but the easy part to say is in bold. Okay, so I just wanted to talk about my background a little bit. So right now I'm getting my master's in biology at UTRGV, but I actually got my bachelor's in sociology from the University of North Texas. I graduated in 2022. And then I did the Texas Master Naturalist Training Program in spring of 2023. That was when I realized that I wanted to change my career. I did not want to be in sociology. It was super depressing. I wanted to play outside. So I worked at Quinta Mazatlan in the education department for a little bit from summer to fall of 23. And then I started the master's program in the spring of 2024 at UTRGV. And then just a little bit about me. I love plants, obviously. I love science. I actually grew up immersed in classical music, and I didn't do anything else until I got to university. I got taught how to play classical guitar when I was 4 years old, and I didn't do anything else until I got to university when I realized I didn't want to be a music major. And so I just tried a bunch of different majors, and then they were like, if you want to graduate on time, you have to pick something. And so then I did sociology. So yeah, but I don't play guitar anymore. I grow plants. I have a little nursery. It's in my parents' backyard, and I've also been native-scaping their yard since 2022. I haven't finished the whole thing because my dad is really attached to his grass, but ever so slowly, I just take out more of it while he's at work, and he gets home and he's like, "Did you change something?" And I'm like, "No, of course not." Yeah, I really love doing that. I love botanizing and naturalizing. I want to see every ecoregion that there is to see in the valley, and I think that I've seen most of them, but definitely not all of them. Some of them are difficult to access. Also, I run the account RGV Naturalist on Instagram. It's not really translated to Facebook very well just because the two social media apps run so differently, but I started that app because I was taking all of these pictures of plants and butterflies, and I just wanted to share them, and then I realized that I could write cool information in the captions also, so I started sharing these things, and people were into it, so I just kept going and going and going. And so I used that account to try and do as much scientific communication that I can do as possible on there, and people seem to like it. Okay, so now getting into the actual presentation. So I'm just gonna, I know a lot of us are familiar with all of this, but I want to recap in case anybody isn't. So the research that I'm doing is taking place in the Rio Grande Valley of South Texas. I have the little map right there that shows our counties where we're situated in Texas, and then also a picture of the characteristic vegetation of the valley. Yes, it varies a lot from place to place, but probably the most common vegetation type is thornscrub, and this picture is at the Martin Refuge. And the context that this research is kind of motivated by is this idea of habitat loss and reforestation. So on the left-hand side is satellite imagery of the valley showing the extent of habitat loss throughout the area, which I know we're all very familiar with. And then on the right-hand side is a map, I think from American Forests, that shows different potential restoration sites around the valley. So things in pink are top potential restoration sites, and then things in dark green are existing thorn scrub patches, things in light green are potential corridors, things like that. And if you're kind of familiar with like the motivation for restoration in the valley, a lot of it has to do with the ocelot. And so the current top potential sites tend to be situated in areas where they already know that ocelots are going through or where they could realistically go through in the near future. So how does restoration look like in the Rio Grande Valley? This is information that I acquired by just talking to a bunch of U.S. Fish and Wildlife people, driving out to random places, people telling me they'd be there at 9 a.m. and they didn't get there until 10:30, and then being like, can you tell me about how this place got reforested? And just hearing people's stories, because it's not like documented super like well on any specific website. So what is the restoration protocol that's practiced in the Rio Grande Valley? So a piece of land is chosen. Usually it's abandoned agricultural land. Any existing vegetation is cleared. So if there's some trees there, like some mesquites and huisaches, those are actually cut down to plant new trees. Thousands of trees and shrubs are planted in rows like a crop. Usually at the start of the cool season, around October is whenever, I think that's when they do Rio reforestation. Sometimes they do spring ones, but at this point, it's starting to be frowned upon because then the plants have to deal with summer almost immediately. The plants are covered with tree tubes. These are about 2 foot long plastic tubes. They look like this in the picture. There are trees inside of all those tubes. And those tubes are to protect from herbivores, and they also provide shade for plants from the sun. Then the plants are left on their own. The tubes are removed after 1 year, and they're recycled onto the next planting. Sometimes between like 1 and 5 years later, there's some invasive species management that's done to control grasses, like guinea grass, buffalo grass, things like that. Usually it's done through herbicide, and sometimes through mowing, but once the plants start to grow up, it gets really hard to take machinery through there, and so it's easier to use herbicide. Sometimes the plants are fertilized whenever they're first planted, but plants almost never get any supplemental water, any additional fertilizer, any supplemental care. They're just put out there, and then that's it. It's survival of the fittest from there. And so that actually ends up giving us a lot of questions that we want to ask about, like which plants survive, why do they survive, things like that. And so what are some reforestation issues? Because it doesn't just go from an empty field with a bunch of trees planted in it to all of a sudden it's a forest. There are some like bumps that plants encounter along the road. One of those things is the establishment time. It takes decades to go from a freshly planted field to thorn scrub. Another one is invasive species. Here, the worst ones at most restoration sites are buffalo grass and guinea grass. Usually, buffalo grass is what colonizes an area whenever it's in full sun. And then once there's a canopy, then guinea grass comes into the understory because it likes the shade. And then also another issue in reforestation is this question of regeneration of the initial cohort of plants that were planted there, because all of these plants have a lifespan. They don't live forever. And so whenever the plants that were initially put into the ground die, we want to make sure that they are regenerating so that it continues to be a forest in the long term. So what do we know so far? Restoration in the valley and beyond. I know these figures look scary. I promise I will explain them. And also, these slides are a little text heavy. I apologize for that, but I'd had a lot of things I needed to say. So a question that we have is, how are reforested tracts doing? And at first glance, sometimes it seems like they're not doing super well. We know that the initial cohort of plants that are put into the ground whenever an area is first restored, that cohort of plants has high mortality rates, which means that many of the plants that are put in the ground are dying. We also know that survivorship rates of the initial cohort are species specific. That means that not all the species that are planted are dying at the same rate. Some of them have really high survival rates, some of them have really low, and a lot of them are in the middle. We also know that some tracts are showing limited recruitment from the initial cohort, and what that means is that the plants that survived and grew up aren't reproducing very well. And what we can see in these two figures right here, both of these actually came out of the lab that I'm in. I didn't do them, it was students before me whose work ended up turning into these figures. The one at the top is looking at biomass of plants, and so that's just like the parts of the plants. And so this is above ground biomass, so like trunk and leaves. And the red bars, again, at the top, it's the biomass of plants that were initially put into the ground whenever it was first restored. The blue bars are plants that recruited, plants that were like, plants that grew whenever the initial cohort started to reproduce. And what you could see is that there's a lot more red than there is blue, because so far in this tract that this data set was gathered from, the initial cohort has not started to reproduce very well. This was done at La Sal del Rey in an area that I think was around 23 years old. And so it's showing that the initial cohort of plants, although they grew up, they're adults now, they don't seem to be reproducing very well, but we don't know why. And then the figure at the bottom is just showing the differential mortality rates on a species-specific basis. So each grouping of bars is a different species, and the bars represent mortality. And so the tall ones are plants that have high mortality rates. The low ones are plants that have low mortality rates. As you could see, there's like 20 different species here, and they all have different mortality rates. So for some more background, this background is not so much about restoration, but it's about the science that goes into making science. So restoration can be thought of as facilitated succession. What is succession? Succession is how plant-dominated ecosystems develop and change over time. No ecosystem is static, ever. Even what you think of as like a climax forest or climax thornscrub, it's still changing all the time. All of those plants have lifespans. They don't live forever. They are dying and reproducing and growing up constantly. And all of this is part of the process of succession. And so succession is a highly relevant ecological mechanism in reforestation. And why is that? It's because reforestation success means that the initial cohort of plants that were put in the ground all grow up, they reproduce, and their babies do the same. And so that means that the succession process is happening both in old growth thorn scrub, but we also want to see it mirrored in reforested areas. And so that means that we can kind of develop like metrics to study reforested areas if we know a little bit about succession in old growth areas. So why is restoration facilitated succession? It's because in natural ecosystems, an empty field would turn into a forest over hundreds of years. And reforestation is just humans attempting to speed up this process. I want to talk a little bit about vegetation development in the Rio Grande Valley and beyond. So like I said, we can figure out how to better study reforested areas if we know how the vegetation that makes them up develops over time. And that hasn't really been done a lot in the valley. However, there is this really cool place in Alice where they've been doing research on vegetation succession for decades, and there's so much information, and the system is very similar to ours. The species that occur there are probably 60% of them are the same, things like Brazil and mesquite and granjeno. And so we can take a lot of information from all of these studies that were done. It's like in Alice, Texas. We can get information from there, even though it wasn't done over here. So what is the desired outcome of reforestation? This is kind of a subjective thing, but from talking to a bunch of people that work at U.S. Fish and Wildlife, Texas Parks and Wildlife, the desired outcome of reforestation in the valley is a closed canopy thorn forest that wildlife can use. And we actually already have a model for this happening, and that's the process of woody encroachment, which is actually what was being researched at the place I was raving about earlier. So what is woody encroachment? It's whenever you have an ecosystem that's a grassland, and then woody plants arrive, they outcompete the grasses, and the ecosystem turns into a woodland, even though technically it's supposed to be a grassland, or it was a grassland over a very extended period of time. And this process has been studied in a nearby ecosystem, like I was talking about. It's around Alice. And the way that it has been described is that mesquite trees first arrive in grassland, and then birds come in and they stop to perch on the mesquite trees. And then as they're hanging out on the branches, they're pooping. And what are they pooping out? Seeds from other plants that they were eating. And so new woody plant species develop in the cozy mesquite understory as they were brought in by the birds, and patches of woodland start to replace grassland. And that's what you could see in this figure. So the lighter areas are patches of grassland. The darker areas are patches of like wooded vegetation. And what's happened over time is that individual clusters of woody plants start to grow, and then eventually they coalesce with each other, and then eventually you have closed canopy woodland. And in this area, it's supposed to be a grassland. And so the idea of woody encroachment is kind of seen negatively, but if you want an empty field to go from an empty field to closed canopy forest, you actually want that process to be happening. Now, it's debatable whether the areas here that are reforested are supposed to be closed canopy thorn scrub or not, and if they were that historically. But if the goal is closed canopy thorn scrub, then this is probably the process it's going to take to get there. And so we're kind of measuring that. So how about that cozy mesquite understory? What exactly about a mesquite understory is cozy? So now we need to talk about facilitation. What is facilitation? That is basically, it could be boiled down to plants helping plants. So life for a plant is stressful. They don't have legs, they can't move. They have to deal with whatever their environment is exactly in the spot that they're in. They're not animals, so they can't just get up and move. And so sometimes growing nearby a bigger plant can help to ameliorate stressful conditions. And this is because shade from a larger plant provides lower temperatures in an area where you have to deal with 100-degree temperatures. It provides increased moisture in a place where it cannot rain for months. And it provides lower light level in places where like the intensity of the UV rays is like really, really intense during the summer. And so shade can actually protect plants from all of these things. So this process, again known as facilitation, it's very common in semi-arid regions. It's been studied in like Arizona, New Mexico, also in like further south in Mexico. It's been studied in like Mediterranean regions of Spain. It's very common in semi-arid regions. And canopies of nurse shrubs are actually havens for seedling establishment in thorn scrub. This is something that you can see if you just go out into thorn scrub, but also it's been studied in papers from Mexico. And in this picture here, it's a classic example of facilitation between two plants. The plant on the bottom is strawberry cactus, and the shrub that is growing above it is blackbrush. Blackbrush facilitates cactuses that are growing underneath it by providing lower temperatures, increased moisture, and lower light levels. And this picture is also from the Martin Refuge. I love that place, and facilitation is so obvious everywhere there. You very rarely see cactuses like this in full sun. They're almost always underneath canopies. So now on to my research questions. That background took a while, and it kind of mirrors like the actual research process, where in order to even ask any questions, I had to read like dozens of papers to figure out like what questions am I even gonna ask anyway. So yeah, this whole presentation kind of like mirrors the process that I went through on my own to get to this point. So I have two research questions, and they are nested within each other. The first one is, how do microhabitats structure woody plant recruitment? And the second one is, how is this relationship shaped by forest successional stage? And I promise I will define all of these things in the coming slides. So question one, how do microhabitats structure woody plant recruitment? What is a microhabitat? It is a small environment within a larger one with distinct environmental conditions. And I have an example right over here. So this is a large landscape, And then right here is a tree. Let's say it's that one. And under here, there's a lot more shade, whereas out over here, it's very open and it's very sunny. What are woody plants? These are just trees and shrubs. I think most people probably know this, but whenever I was writing the description for this presentation, I checked it with my musician boyfriend. I made him read it, and I said, "Tell me every word you don't know." And he was like, "What's a woody plant?" And so I said, okay, yeah, it's trees and shrubs. I just had to make sure that it was like friendly for non-science people, because once you're immersed in this for a while, words like microhabitat just become part of your vocabulary, and you're like, what do you mean you don't know what a microhabitat is? So yeah, defining your terms is really important. So yeah, I know I kind of glazed over it. Woody plants are just trees and shrubs, as opposed to like grasses and herbs, things like that. And what is recruitment? Recruitment is plant regeneration. Basically, germination plus establishment and or death equals plant recruitment. And these are heartleaf hibiscus seedlings that I found in the field. So this is still kind of defining the first question. I already defined what a microhabitat is, but which microhabitats are we using? So we have 3 different types of microhabitats. The first one is canopy cover, which is in the shade. One of them is canopies of Texas ebony. Texas ebonies have a very dense canopy. They provide deep shade. And then our other shaded microhabitat is mesquites. They have a sparser canopy, and so it's more like medium shade. And then we have our full sun microhabitats, which are basically just the spaces between the trees out in like whatever sites we're at. And these areas are in full sun. There's no shade. Now on to question 2. How is this relationship that we talked about earlier, the microhabitat question, shaped by forest successional stage? So I'm using this term forest successional stage to basically mean the age of the forest. And we have 2 different stages. One of them is undisturbed. These are habitats that we suspect have not undergone human-generated disturbance for 100+ years. And I really had to qualify that, because whenever you say old growth, that has a lot of connotations that go along with it. And in the valley, we actually don't really have a way of knowing what's old growth and what's not, because everything here has been so disturbed for so long that it's kind of impossible to know, like, well, what if this area was cut down a long time ago? It's very difficult to know. And so instead, we call the areas undisturbed, because humans haven't gone and bothered them for at least 100 years. And we can determine that by looking at satellite imagery. There's satellite imagery going back to the 30s, and if we can see like a lot of trees in a place in the 1930s, that means that it's been growing for at least 100 years from now. Our second successional stage is reforested ones, and these are habitats that have undergone active restoration. So what are the hypotheses for this research? The first one is that canopies of ebonies and mesquites act as key sites for regeneration relative to open areas. So we think that plant recruitment is going to be happening more and better underneath canopies compared to out in the open. And then also, older forests, so the undisturbed ones, should have a denser and more diverse understory than the younger ones. So I'm going to take a couple minutes to talk about my study design. In this figure, I have the site layout and our microhabitat types. So we have 4 different sites that we're at. Again, 2 of them are reforested, 2 of them are undisturbed, but among the reforested ones, the one up here has a lower planting density, which you can see because the trees are further apart from each other, and the one down here has a higher planting density. You could see that because the trees are closer together. And what I mean by planting density is the way that whoever does the reforestation, it used to be U.S. Fish and Wildlife, now I think it's going to be American Forests, but the way that they would decide how many plants are going to go out there, they wouldn't just like randomly take a bunch of plants and then put them out there. You decide beforehand how many plants per acre. And right now, the density that they plant with is about 1,000 plants per acre. But back in that like first tile of satellite imagery, they were doing 100 plants per acre. And then the one below it, they did a little bit more. And so whenever these places were first reforested, some of them had more plants that were put in, and some of them had less. So that is for our reforested successional stage. For undisturbed, there's 2 different sites. One of them is the Schleben tract. This is a tract that's off of 186 in northeastern Hidalgo County. It's really close to La Sal del Rey. Schleben is not as xeric as you'd expect a lot of thorn scrub to be. The trees are pretty tall for being in Hidalgo County, at least in northern Hidalgo County, and it's not super diverse. And then we also have the Martin Refuge as our second undisturbed site. The Martin Refuge is more xeric, so it means it's drier. The vegetation is shorter. It's about 1 meter to 2 meters shorter than the vegetation at the Schleben tract. And it's also more diverse. It's a lot more diverse. There's like a new plant every step that you take, there's a new plant. And so the Schleben tract and Martin Refuge are the 2 undisturbed sites, and the 2 reforested sites, they're both at La Sal del Rey, but that tract is really huge, and different parts of it were restored at different times. And so some of them are denser and some of them are sparser. And then just to recap on microhabitat, there's ebony trees, the canopies of ebony trees, which provide a thick overstory, the canopies of mesquite trees, which provide a thin overstory, and full sun, which I usually refer to as open areas. And then just a quick site/sampling breakdown, because I don't just go out there and look at plants. We have like a specific protocol that we use to go out there. So here I have an example of a single landscape at a specific site. This is at the Schleben tract. So all of this would be considered one landscape, and then we need to have replicates of plots that we do. And here I have an example of 3 different blocks, and each of these blocks has 3 different microhabitats in it. So right here I have an ebony tree, a mesquite tree, and an open area. That's one block. Right here I have an ebony tree, mesquite, open area. That's another block. And same thing right over here. And we do this because in science you need a lot of repetition to make sure that the things that you're seeing are happening for a reason and it's not just random. And our 3 other landscapes can be broken down similarly. Our total replicates are 39. What are my methods whenever I go out there? So all woody plants within a plot are measured and tagged. Here's a picture of the 2000th plant that I tagged. It's a guayacán. I was really happy that it was a guayacán and not a colima, because those are really mean to you. What's the data that we record? So I take down the species. We have a coordinate system within our plots so that we know which plants are in which parts of the plots. Ebonies and mesquites can actually get pretty big. We have some plots that are over 10 by 10 meters. Like as a square. And so it's helpful to know where in the plot the plants are. And so we have a coordinate system within each plot. Also the height of the plant and the stem diameter. So if you're at all familiar with forestry methods in places that are not the valley, there is a concept of diameter at breast height, and it's because trees actually look like trees, not shrubs. And so if you want to know the diameter of like the size of the trunk, you could just go and measure it like right here. Our plants, you can't do that because they branch really low to the ground. And so in our lab, we take diameter at ground height. And so we get on the ground and we take the diameter of the plant right here. And sometimes, actually most of the time, we have to like dig down a couple centimeters to find where the 2 branches of the plant actually like coalesce together. So some very preliminary results, there's more later, but I just like bragging about how many plants I've tagged. So we've surveyed 39 plots covering over 2,000 square meters at 4 different sites. We have measured over 2,800 plants and tagged every single one. And we've encountered 43, I think now it's 44 unique woody species, because I was confusing wolfberry for snake eyes for the longest time, but now I separated them. And in this picture, it's one of our plots with the first tags that we ever used. They were huge. They were like the size of my hand, and they were like these like plastic orange tags. And I learned really quickly that when they're in full sun, they will melt. And so I had to replace every single plastic tag with metal ones, which we're actually still working on right now. That's pretty fun. But now the pictures look different in a plot that's like filled with tags. So now getting into the meat of my results. So there's several different angles that we took to analyze the data that we got, because I have this gigantic Excel sheet with like 3,000 rows, and every row is a plant, and like, what do you even do with that? There's a lot of things that you can do with that if you know statistics. So one of the questions that we have is, does plant abundance vary by microhabitat and by successional stage? So is there different levels of plant abundance under ebonies, under mesquites, in open areas, and does that also change in undisturbed areas compared to reforested ones? And so what this figure shows is that canopies support higher plant abundance than open areas. Undisturbed sites support higher plant abundance than reforested ones, and abundance refers to density. And what does density refer to? That's plants per unit area. So imagine different amounts of plants within a 1 meter square area. Like if you could draw like 1 square meter right over here, imagine there's 1 plant in it, then the density is going to be 1, because it's the amount of plants divided by the area of the square. But 1 square here might have 1 plant in it, and another square over there might actually have 10 plants in it. And so plant density can change throughout the landscape. And so we're looking at how that density changes based on the different microhabitats and the different successional stages. And so what we're seeing is that the old growth undisturbed sites are a lot denser than the reforested sites, which is like, duh, you would expect that, right? You would hope that an undisturbed site is denser than a reforested site. But also what we're seeing is that underneath canopies of ebonies and mesquite, there's a lot higher plant density than out in open areas. So does plant abundance vary by microhabitat and successional stage? Clearly, yes. Undisturbed landscapes are more abundant than reforested ones, and canopies are more abundant than open areas. More plants per unit area equals more dense. Also, what does this mean? So higher stem density equals more cover, which equals more animal usage. More cover means lower invasive species pressure, which means there's little to no grass. So denser forests are going to support more usage of the forest by animals, especially target animals like ocelots, which like to crawl around in like thornscrub. And also these denser areas that have very thick canopy cover are going to have little to no grass underneath them. So another question we have is, does species richness vary by microhabitat and successional stage? What is species richness? That's just the number of species in an area. So let's say you have an area that's the size of this room, and it's filled with plants, but it's all just 1 species. Then that area, the species richness is very low, because there's only 1 species in here. But this same area in undisturbed thorn scrub could have like 50 different species. That would be higher species richness, because there's more species in the same area. And so what we see here is that undisturbed sites have way higher species richness than the reforested ones. And also, canopies have higher species richness than open areas. And again, for density, it's plants per unit area. And imagine different, so before I talked about imagining like a 1 square meter area with different amounts of plants in it. Now you can imagine that same area, but with different amounts of species in it. So it can have 1 species, you can have 2, you can have 3. And that species richness that number is gonna change depending on how many plants are inside of that little square. And so that's how we come up with these calculations. So does species richness vary by microhabitat and successional stage? Clearly, yes. More species occur in undisturbed landscapes than reforested ones, which is, again, duh, you would hope that undisturbed landscapes would have more species than reforested ones, but we just want to check. And also, canopies support more species than open areas. And going back to this slide, what we can actually see that is very important here is that, yes, the blue box plots that are referring to undisturbed sites are bigger, and like the black line refers to the median, and that's higher than in the reforested sites, which shows that both reforested sites don't have as many species in them as the undisturbed sites. However, this would be the median for undisturbed sites in open areas. And this is the exact same thing, but in subcanopies of reforested areas. And what that means is that under canopies within these reforested areas, it's actually starting to resemble the same amount of species richness that would occur in undisturbed sites in the open. And so whenever these numbers start to approximate each other, we could take that as signs that these reforested areas are actually developing on a good trajectory. It doesn't mean that they're the same thing. It also doesn't mean that they're ever going to be the same thing, at least not in any of our lifetimes. But it does tell us that some of these reforested tracts might actually be doing better than we thought they were. Also, what is the implications of all this species richness stuff? So higher species richness means more plant biodiversity, and more plant biodiversity is linked to greater biodiversity in general. Generally, more types of plants means more types of animals. So the more plants you have, then the more butterflies that you can have that can host on the plants. And the more variety of butterflies that there are, and the higher number of butterflies that there are, then you get more birds that are coming in to eat the butterflies. And so it kind of just like goes up from there in trophic levels. That's why having high species richness is important, and biodiversity in general. So this is a kind of a different line of analysis now. Are there species-specific preferences or patterns based on microhabitat? And what we can see is that species don't occur equally or evenly throughout a landscape. They have preferences for specific microhabitats. I have, I think, 9 different species on this list. And the different bars in different colors are, they correspond to different types of microhabitats. So the green bars are under ebony canopies, the blue bars are under mesquite canopies, and the purple ones are in open areas. And then the red one is just like an all category. And what you can see is that, for example, snake eyes at the very bottom really, really likes ebony canopies. There's like so much of it in ebony canopies. Whereas desert yaupan, which is a little bit above the snake eyes, actually really prefers mesquite instead of ebonies. So the plants are not occurring, like, they're not just randomly distributed throughout the landscape. They occur in specific areas. So are there species-specific preferences or patterns that are based on successional stage? We could see that species preferences for microhabitat actually change across different landscapes. And so on the left-hand side, this is those species preferences we were looking at earlier, but in reforested landscapes. On the right-hand side is those same species preferences, but in undisturbed landscapes. You could see on the right-hand side for Coma, the first one, it prefers open areas, whereas in reforested areas, it prefers ebonies. And there's so many different reasons for this, but it is interesting to look at how like the different species are arranged throughout a landscape and if their preferences change depending on like what type of landscape they're in. So this is a totally different line of analysis now that I'm going to explain a little bit. So you know how I talked about how we collect plant height and also diameter? Well, we do that for a reason. That's because we can actually learn a lot of information about vegetation if we know the sizes of plants. And so these graphs are all size class distributions. So the distributions of plant size can tell us really interesting things about their populations. If you think about it, if you go into a forest and you're looking at ebony trees, they're not all at the same size. Like, it's not an orchard. You have ebonies that are really big, you have some that are medium-sized, and you also have seedlings and saplings that are growing everywhere. And if you know what the size distributions are, like how many seedlings are here, how many saplings are here, how many adult trees are here, that can actually tell you information about what the ebony population is going to look like in a couple decades. So a reverse J shape in the bar chart of plant size can indicate that the population is reproducing well. When I say reverse J, what I mean is that it's shaped like this, and a J is shaped like that. So if you take the J and just flip it on its side, then this is reverse J shaped, as you can see right here. I was supposed to put animations on this, but I ran out of time. That would have made it easier. So Again, this reverse J shape in the size class distribution of the plant can indicate that the population is reproducing well. And that's because you're supposed to have a lot of seedlings of a plant, and then a bunch of those are gonna die. And of the ones that make it, they're gonna grow into like teenager plants. And then a bunch of those are gonna die, and the ones that make it are going to become adult plants. And so the thing that makes the most sense is for there to be a ton of seedlings, and then less saplings, and then even less adults. But other shapes of this distribution are not as promising as that reverse J shape, and that's something that we'll see in some of the following slides. So how does microhabitat affect the distribution of plant size? Like, if you look at this, this is not separated by microhabitat, by site, by successional stage, anything like that. This is just all of the data aggregated to look at the size class distributions of granjeno, cenizo, things like that. But here, it's separated by microhabitat. There's 3 different graphs. The one at the top left is ebony, bottom left is mesquite, and then the one that's kind of on its own is open areas. And we're comparing 4 different species: granjeno, guayacán, desert yaupan, and coma. And what you could see for the first one, granjeno, which is in red, it's that it has the reverse J shape for ebony microhabitats and for mesquites, but in open areas, it's not reverse J, it's just like a J shape. And that can indicate that the plant is not reproducing very well in these areas. And the same thing is the case for guayacán. In ebony and mesquite habitats, it has that reverse J, but in open areas, it has that regular J shape, which in ecology is not necessarily a good thing. But desert yaupan is really interesting because it has a really strong reverse J in every single microhabitat. And there's like a ton of different reasons for that. We don't know exactly why, but it's interesting seeing how this stuff changes from species to species. And then coma is also interesting. So it has a very slight reverse J under ebony microhabitats. And then in mesquite, it's like, I don't even know what it's doing. It's just kind of random. And then in open areas, it has the J shape. And so basically, if the size distribution of a species in a certain area is not the reverse J, then that can have implications for whether that plant is going to remain in the community over the long term. And when I say over the long term, I mean like decades and also like hundreds of years. Okay, now I know that these look like size class distributions, but they're not. It's something different. So this is plant density, so how many plants are in that 1 meter square. And it's mapped as distance from a host canopy tree. So if I'm an ebony tree and there's a bunch of plants around me, do they maintain the same density as they get further away from me? Is there more plants that are closer to me, or more plants that are like a meter away from me, or more plants that are further, or is it the other way around? Is there more plants closer to me and then less plants further away? And this can tell us things like this idea of like a nurse plant Goldilocks zone. So if an ebony tree is functioning as a nurse plant, where do the plants that are its proteges like to hang out? Is there a Goldilocks zone underneath this nurse plant? Is it really close to the trunk? Is it really far from the trunk? Or is it somewhere in the middle? And then if we can determine where that zone is, we can ask why. Is it because of nutrients that are under the canopy? Is it because of light levels that are underneath the canopy? Probably if you're standing super close to the trunk of a tree, there's more shade than if you're standing far, like at the end of the canopy. And so this is done on a plot-specific basis, so every single one of these graphs represents one specific tree. It's a mixture of ebonies and mesquites, and we're actually seeing the exact same pattern, just at different like levels, and the curve is like steeper in some and then lower in others. But the highest densities of plants tend to be kind of close to the tree trunk, but like not right against it, which makes sense. I mean, plants probably want space to breathe. And so, yeah, we're seeing that the majority of plants in these plots are closer to the trunk rather than farther away. Yeah, and so this can tell us about this idea of like a Goldilocks zone near the plant. Keep in mind, something that I hadn't brought up earlier is that the surveys that we've done, they're not done yet. And so this data is looking at everything that we have, but we haven't finished everything. And so a lot of these numbers are gonna change every time. And actually, the place where I make these graphs, it's in a program called R. It's a programming language. It's basically like computer coding, and it's really hard, and I kind of hate it, but I also love it. But it makes it so that I can write lines to tell the computer, make a graph out of these numbers, and every time that I run those lines again on the computer when I have new data, it just remakes these graphs. I don't have to remake them. All I have to do is hit Control-Enter, and then it redoes them whenever I get new data. And so by the time we finish all of these canopies, a lot of these distributions are probably going to look different. So that gets us back to the first question that we started off with: how does a forest sustain itself? So microhabitats created by tree canopies seem to be essential for woody understory development. In reforested areas, more recruitment is happening under canopies than out in the open. This is important because probably a baseline assumption would be that plants are recruiting evenly throughout a landscape, or they're recruiting randomly throughout a landscape. But that's not the case. They seem to prefer these shaded microhabitats compared to out in the open. Species also clearly have specific associations with different microhabitats, and they don't occur uniformly throughout the forest landscape. Most species prefer shaded microhabitats, some prefer open, and there are a few generalists that could live anywhere that they want, but most of them have a preference for a specific type of microhabitat. And then also, based on our size class analyses, many species are showing promising signs of future recruitment, which in an undisturbed area, it's like, okay, whatever. If granjero's not gonna be here in 20 years, I mean, who cares? There's Brazil, the birds will eat that. But in a reforested area, it's actually really important to see if the species that you initially planted there are showing signs of future recruitment, because it shows like which ones are doing a good job and which ones aren't. And it also shows like plant competitive ability, stuff like that. What are the implications for reforestation? So a one and done planting method for all different species may not be the best approach, and that's currently what's done right now. Every single species that they want to include in a reforestation is just planted out into the field, and it's done randomly. They're not grouped together in any specific way. And this method may not be the best approach if you want to increase survivorship as much as possible. So augmentation planting, I'll explain that in a second, utilizing mature canopies is worth exploring to increase stem density and species richness. Augmentation planting, I actually forgot to define my term here, is the idea that you would return to a reforested site after it's been reforested and add in new plants to help develop the populations. And it could be by going in and like physically planting seedlings. It could also just be through direct seeding, like just going out there and spreading the seeds out there of plants. And that's actually a method that's done commonly. It's just difficult to do if you're doing restoration in like a mechanized way, because everything costs a lot of money. These reforested areas are really huge, all of that stuff. However, certain species do thrive in full sun. Not all species want to be under the canopy. Like Cenizo really likes being in full sun. And understanding and utilizing these species traits may be key for reforestation. Like, for example, if you want to plant out a bunch of different species in this open field, but you know that snake eyes really doesn't like being in full sun, then if you go and plant 1,000 snake eyes in this field and only 2 of them survive, you just killed 998 snake eyes and also wasted all the money that you spent to buy those 1,000 snake eyes. What if they could be introduced at a later time when they are more likely to survive? Oh, I guess that's the end of my presentation. So some future work, just so you guys know what I'm working on, what I'm gonna do with this. So I'm going to continue collecting data. Me and also all of the people that help me with fieldwork, we have to finish tagging the plants. We have to collect soil samples. I am working on writing and publishing a manuscript so I could have a paper in a scientific journal by the time I get out of graduate school, which is the goal. And also, I am working on collaborating on analysis of soil samples for nutrients, specifically carbon and nitrogen. I didn't include any of that information here because it's a totally different topic, but we have samples from all of our different microhabitats, and it can be interesting to look at, like, we see differences in vegetation, but are there differences in the soil that are mirroring those vegetation changes, or are there not? And if there's not differences, one of the things that could mean is that the plants actually don't really care about differences between soil under canopies versus out in the open. What they really care about is the shade. It could tell us things like that. And here are my acknowledgements. Thank you so much to all of the people who are on here. I think most of these people, actually, you know what? A lot of these people are here. Haley is here. She's my fieldwork assistant. She helps me with literally everything. She's amazing. Josue is here. He helped me with a ton of fieldwork. Sophia's not here. Lorna is right here. Lorna helps me with fieldwork all the time. She's fantastic. Aaron is here. He helps me with fieldwork. And then also, one of the sites that I kept mentioning over and over again, it's the Martin Refuge. Thank you so much to John and Audrey. I don't think they're here, but Audrey told me she would watch the YouTube version of this. So thank you, Audrey, for giving me access to your amazing land. Thank you to American Forest and UTRGV. And that's it. Yes, I can answer questions if people have them. Yes?
Yes, is there a difference in the plant mortality in a restoration project that begins on new agricultural land versus one that starts on land that was never in agriculture?
So that's not something that we've looked at, but I would say that probably the differences in mortality, if they're affected by the soil, it's gonna have less to do with like if there was agriculture and more if there was any soil disturbance to begin with. And so I don't know, like I know that here it's mostly agricultural land that's reforested, but in other parts of the world there's like abandoned mining land that's restored, and it probably those areas probably struggle with similar issues. But yeah, I think that it probably has to do with the level of disturbance that the soil had to deal with. But here we don't have like specific comparisons that we can do, especially because like I don't know if there's any land that wasn't agriculture that U.S. Fish and Wildlife has restored. Maybe there is, maybe like old ranching land or stuff like that, but places that have buildings on them probably still have buildings on them. So yeah. What was it? I think it was if there's differences in mortality for restoration sites that were done on old agricultural land compared to other types of land? Okay. Yes?
The Fish and Wildlife revegetation here, they varied over the years from 100 seedlings per acre to 10,000 seedlings per acre. What does your research shed any thoughts on the density of planting?
So that's not something specifically that we've looked at. I actually, I've visited way more sites than I actually have plots on, and one of the sites that I visited was at Laguna Rascosa, where they had like a pretty small area and like an overabundance of plants, and they didn't know where else to put them, so they were just like, here, put them all here. And so the plants are super close together. Like in most places, the plants are like, I don't know, like that far apart, but at this spot, they're so close together. The density is so high, and we're not studying that area because it would be too much. But I would assume that the higher planting density means that whatever is there, the survivors, there's going to be more of them, and so it's probably going to be denser compared to areas where it was 100 plants per acre. That would be my assumption, but we're not directly testing that. Yes?
Question, how would you take your research and apply it to people who are not doing a reforestation project, but doing a rehab? We have a Italian manual, where we're rehabbing, we're taking out all the invasive kitty grass. But we don't want it to look like a garden, we want it to look like the original lawn forest. What would you recommend to achieve?
Okay, I'm going to repeat the question so people can hear it. how could this research be applied to people who are not doing restorations, but instead they're doing more like land rehabilitation, specifically in like wild areas where they want it to resemble brush? I would say that type of like doing those activities on the land, that counts as augmentation, because you're augmenting what's on the land. And so I think that it could follow like a pretty similar method as the restorations. I know lots of people here have done like private restorations on smaller tracts of land compared to like 10,000 acres. And there are similar things that you do. I would think that if you're going to be working on a smaller piece of land, it might be helpful to not plant in rows, but instead to kind of like plant a little bit more randomly. Like, it's not going to be random because you're the one doing it, but so that it doesn't look like an orchard. Like some of these areas, whenever you walk in, it looks like orchard rows of ebony trees, and that doesn't look like brush. But yeah, I think that would count as like augmentation planting. So basically, like, you take note of what's there, and then you think, well, what do I want to be there, and what do I not want to be there? Which just sounds like that's already what you're doing. And so I think that the findings of this are probably going to be similar. Like stuff that you plant out underneath canopies is probably going to do better than stuff out in the open, unless it's specifically plants that want to be in full sun. But also, if you're doing like a small scale thing and you're like supplementing with water and fertilizer, that completely changes everything. Like the plants are going to be so much happier. I have guaiacán in full sun at my house. I have snake eyes in full sun at my house, but I water it. If I didn't water it, it would be dead. Yes?
So I'm kind of wondering about, as far as how successful a rewilding is comparative, if you, like you talked about with the Rio reforestation, a lot of times it just randomly just blitz it with all kinds of different species. I wonder if there's been any kind of study of comparing that method with having a higher intensity of keystone species that are known to bring in more insects, which will bring in more birds, and will allow things to reseed a lot more quickly.
Mm-hmm.
You know, comparing those two methods against each other.
Okay. So reforestation, I'm repeating the question. Reforestation, where you kind of like randomly distribute all of these different species throughout an area, compared to selecting specific species for a specific reason that you can put out in areas. So that was actually something that I kind of skipped over because the text was small. So in this slide, I don't know if you could see at the bottom, it says, aligns with previous research on trait-based reforestation approaches. And I didn't translate that into regular person words because it's too much to talk about. But basically, there's this idea of plant functional traits, which is where you can group plants by their traits instead of necessarily by their taxonomy. So instead of saying, I'm going to group everything in the legume family together and just plant all of those out there, instead you're going to be like, okay, well, these plants have thorns, these plants have pinnately compound leaves, these plants like being in the sun, these plants like being in the shade. And the question that you asked, like plants that will attract more biodiversity, you can kind of group them into a functional group. Like, we want these species to be out here because these attract bees. Well, now you just made a functional group of species that attract bees, or species that attract butterflies, things like that, or species that are going to reseed really well. And there actually is like reforestation research that's being done on that, where they don't just randomly select, like, we know that, let's just say, I know there's more, but let's say there's 30 shrubs that are native to this area, and we're going to take all 30 of those and just randomly distribute them all throughout a field. Instead, you could be like, well, yeah, all 30 of these are native, but 10 of these tend to do really well, so we're only going to put those. Yeah, studies like that are being done all the time. Yes?
In the mid-valley, these Tippawahis are pretty common, early succession tree. Grows fast, but you haven't mentioned that at all. Is that something?
That's about tepeguajes, how they're an early successional tree and they grow really fast. Are you asking like if we looked at that specific species, or if something like a similar thing has been done?
Tree, whether you looked at that.
Sorry?
In terms of the understory under Tippawahis, did you look
Oh, no. Specifically because the UTRGV Edinburgh campus is like a little bit further northwest than where a lot of the tepeguajes are. and we already had our sites determined. A lot of the stuff that happens in terms of study design isn't because like, oh, we have like the perfect design, we have the perfect sites that we're going to choose. It's more like, where can we afford to drive to? It's things like that. And I have been to sites, like I went to Southmost in Brownsville, that whenever they did the restorations, there's a ton of tepeguaje, but we realized that the sites would be really far, and so it was more difficult to do. And then also, because our microhabitats were ebony, mesquite, and open areas, we kind of had to stick to landscapes that had all three of those species. And whenever we went like to Southmost, we went to Sable Palms also, we were like, where are the ebonies? There's a couple of them, but then there's not mesquites next to them. And so we couldn't make the design in the exact same way. So no, we haven't looked at that.